I'm a PI in mechanical engineering, and Dino, who's the speaker today, he just joined UCLA a few months ago in fall of 2020. So Dino started his journey into science through a PhD in physics at University College London. And then he moved to, for a few years, to the University of Bar Ilan in Israel. During his PhD, he studied the nuclear pore complex as a modeler. And then he moved on to his postdoc, where he continued to work on that, but also he pursued work on polymer chains, modeling polymer chains. And then more recently, as an independent fellow at MIT through the Living Systems Fellowship, he started working on phase separation and droplets, which is what then brought him to UCLA. My group is interested in designing artificial molecular droplets with nucleic acids. And so Dino is supporting us by building predictive models of how phase separation with RNA exclusively could work out. So with that, maybe I will also let Dino say a few words about his own journey. So why don't you take
thank you.
From here.
So I'll just share my screen. So I hope everyone can see this. Yes, so actually I think if I talk about my kind of the personal aspect of my background might be good contextualization for the talk. So, and my career is, or at least not to call it my career, but kind of my curiosities have followed a somewhat of a boomerang trajectory. So when I was young, I was really interested in animals, nature documentaries, that kind of stuff. And then when I was about 11 or 12, and I first started reading about evolution, I was kind of very taken with the kind of being able to have some kind of generality to things that you see in a way that kind of I hadn't anticipated or something like that. So I've always been kind of very interested in generality. And as you might expect for someone who's kind of interested in generality, they might start to, biology is kind of very difficult and messy and tricky. And so someone interested in generality, you might expect to find in physics, which is what I did. So then I was kind of in physics for a while, and I was maybe more interested in kind of very theoretical issues. But then I've kind of slowly since then have been arcing back. I won't say that I'm fully back into being anything like a biologist at the moment. So I'm still very much a physicist, but I've been kind of more interested in describing biological phenomena throughout my career. And that's kind of been the guiding light, but in a way that tries to keep it in a way that a physicist might like. So in a kind of generic way. So what is there kind of generic to say about biology? It's kind of difficult, but biology does have different kinds of challenges which it has to address, and most of my work has been focused inside cells. And one kind of approach to which you can think about cells is they're really having to, especially eukaryotic cells, really having to organize their chemistry in some way. So Eliza mentioned that I worked on the nuclear pore complex, which is one example of a cellular mechanism which is organizing different components across different compartments to organize chemistry. And in general, for a eukaryotic cell, you need to be able to have all of these different chemical components inside to have the correct reactions and so on. So especially kind of in a cytoplasmic context, if you're trying to find or trying to realize a specific kind of chemical reaction, you have a problem akin to trying to find a person in a crowd. The cytoplasmic environment is dense, and there's lots of different kinds of proteins and molecules in there. And so one question that I kind of started thinking about prior to phase separation, actually, which is how are robust protein-protein interactions realized in this sea of non-specific interactions? So how do I find a particular partner in a crowd? Now, for eukaryotes, there's several mechanisms that I'm sure you must, people who have done biology are familiar with. So one thing, a definition almost of eukaryotes, are the existence of these membrane-bound organelles, which are necessary for organizing these different chemical reactions in space. And here you can see an electron micrograph of such. You can see some of these membrane-bound organelles. And without them, without these compartments, the cells rapidly degrade and die without having the ability to control their chemistry. And this has kind of been a field of study for a long while. So I think people have observed these compartments, these membrane-bound compartments for a very long time. And as I'm sure you're familiar, in recent years, there's been kind of a more broader interest, or there's been this kind of new interest in trying in membraneless compartments, which is, you can see on the bottom kind of one of the papers that kicked off this idea with P granules. So instead of having a membrane surrounding these membrane-bound compartments, it's possible through physical mechanisms to form compartments via liquid phase separation. And this is kind of where you might also consider that someone on the physicsy kind of approach might be interested, because it does have the kind of the mechanisms of phase separation have been well understood in physics for a long time. And now in the past 10 years, people have been thinking about them more in the context of biology. And so there's also at the top some review articles around this. And a lot of effort has been now spent on attempting to find these different phase separating systems. And my perspective on this is kind of, it's, what would I expect kind of this dense interacting soup to have in general? You might expect phase separation just because of the nature of that kind of liquid, which is the cytoplasm. But it's been harder so far, as far as I'm going to really think about the possible functional consequences of having membraneless or liquid-liquid phase separation. So, and this is from a review article a few years ago. Here are kind of some of the mechanisms, some of the scenarios which people have gone over in terms of how they think having this mechanism of liquid phase separation might be useful to the cell. And the net result basically of all these different scenarios is something familiar to what I was talking about with regards to membrane-bound compartments, where having some control of chemistry is important. So the idea being that you can also control chemistry via this mechanism. So obviously, in the case where there's no membrane, it would be much easier for things to kind of transition across that membrane. So, for example, if you have two different kinds of molecules and you concentrate them within a liquid droplet, then that will increase reaction specificity and lead to an increase in product that the cell might be interested in. And conversely, you can kind of, by tuning the properties of these droplets, you might be able to tune the chemical makeup of the cell in response to particular stimuli. So that's kind of the scenarios here, A, B, and C. And then another mechanism is by having these droplets, you can kind of buffer the reaction rates against stochastic fluctuations in production of genes. And so these are kind of postulated mechanisms of how phase separation can help with controlling chemistry. And so that's kind of where I became interested in this problem. So actually, with regards to what I was saying before, I was actually thinking about something very different, which is kind of statistical physics, when I have every particle in my system is of a different kind, which is kind of another problem. And then I started to think about kind of simple models for what I call complex chemistry, which means chemistry where there are many, many different components, not just two. So usually on the theoretical level, when we're kind of describing things, we're usually considering things in only one or two, three different components, because the complexity of the problem starts to kind of explode as the more things you have. So I kind of, with respect to that, I was thinking about that problem and then thought about this kind of separate problem. So if I have say a system, now we've kind of jumped from the biological motivation to kind of more theoretical questions, which is is that mechanism, are those postulates that people came up with reasonable in terms of kind of simplified models? So let's say I have a very large system with many different kinds of particle in there representing who knows what inside the cell, many different kinds of protein. And now I have, say, a specific subset of those particles which are that you want to be realized, or you want those reactions to occur. But you also have non-specific reactions of a different kind, and those vastly outnumber the specific kind. So that's why. So here I've drawn a graph of kind of the probability of these finding different energy scales in the system. So let's say I have many kinds of interactions which are about a kT for forming something which is kind of some non-specific interaction. And then I have many less, which are what I call good agglomerate, which have a higher energy, and I kind of represent that in terms of this set at the bottom, where you have kind of within a set you have different kinds of you have this kind of good agglomerate, and then every particle outside of its set has this bad agglomerate. And the question one may ask is, how did the non-specific interactions change the output of the specific interactions? So, and this is something that I asked before being aware of the kind of experimental nature of phase separation, actually. So, but what you see and what you can see in this figure is that, say for example, I have this same chemical, this mixture of many different kinds of chemicals, and I look at the ratio of this good product that I form as a function of the non-specific interaction. One can see, and this is what I'm plotting in this graph. So as I go beyond a particular point, which is the formation of droplets, which in this one is about 1.4 kT, so non-specific interaction, then condensation into droplets helps you find, or helps the formation of these specific interactions greatly by reducing the search space of possible, by reducing the search space that each specific pair has to go through in order to find that kind of good reactant that you would like.
I'm sorry to interrupt, Dino. You may have addressed this earlier. I had to be late because I had an earlier meeting that went a little. I'm Eric Dietz. I was just curious, what kind of simulation are you running here? This is a Brownian dynamics, Langevin dynamics.
Yes, so this is Brownian dynamics with,
and the interactions are isotropic, meaning
yes,
that
interactions are isotropic. So what you have is kind of every particle is isotropic, so the valency is effectively however many particles you can fit around you. So and then there's, within the system, you kind of define a subset of these particles which have this greater interaction energy.
Yeah, and the potential is something like a van der Waals, like a LJ 612.
Van der Waals, yes.
Cool,
yeah.
Cool, thanks. Those details may not be important, but I just wanted to understand.
No, that's great. So, yeah, so if you're interested, then this paper was published a few years ago, and I go into these details. In general, though, kind of the what's going on is I have all of these particles when they're kind of in this gaseous phase, it's hard for there's a much bigger space to search in order to find a particular partner or to form this good product. As I condense, then you don't, and it's still a liquid, then you have a much smaller space in which to search in order to realize these chemical reactions. But then as I make the non-specific interaction, or this multivalent, if you want to think about it that way, interaction much larger, the system starts kind of, then you actually start competing with these specific interactions and you start lessening them, which is why this graph has a kind of a transition to the formation of product at the formation of when you start to see droplets, and then gradual tapering off as you increase the strength of the non-specific interaction. So that's kind of, this is a very kind of simplified idea about how condensation might be able to help with regards to these problems of forming reactions. But there's some issues with this framework, which is kind of very non-biological. So one thing is that if I have kind of if I have something that forms a droplet, it's not really, the problem kind of gets worse as the droplet gets bigger. If I only have, let's say I have 5 particles inside this droplet to the left that are forming this good product. So those might be proteins or whatever. As I scale up the droplet and there's still only that same number of particles, the amount of kind of this entropic effect, the amount of these non-specific interactions goes as n squared, so the size of the droplet squared. So even if I were to increase the number of particles within the droplet in a commensurate way, big droplets are kind of less helpful at organizing chemistry than these small ones. But there's another problem, which is that in at least an equilibrium theory or a physical theory, if I have many droplets in a system, they have a tendency after time to start ripening. So that's kind of in this small diagram at the bottom. If I have these droplets and I just come back to them at a later time, I would eventually see everything in a big droplet. But small droplets are more helpful than big ones. And that's kind of a question with regards to the ordinary literature on phase separation. Is if you see many different droplets, it's either kind of maybe a very slowly relaxing state. It's not a fully equilibrated state, or it could be that those droplets are being actively maintained somehow. So that's kind of been something that physicists have been thinking about recently, which is I have that kind of simplified scheme of phase separation and chemical reactions, but in reality, these reactions might not be in equilibrium. So there might be some energetic input that's necessary for some of these reactions, and in biology, that's definitely the case because all biological systems need some kind of need to consume some kind of energy from the environment. So yeah, this has kind of spurred some interest in recent literature. People have called this kind of an active emulsion. So let's say I have a system with many different kinds of droplets, but the droplets are not, and usually we call that an emulsion. So it's stabilized by surfactant or something else. But what if they're being stabilized by non-equilibriumness somehow? So some kind of energy input. So if you're interested in that, this review article goes into it in some depth. But people have been seeing some interesting results when you kind of break out of this equilibrium condition, which I was discussing previously. For example, if you have a droplet with non-equilibrium chemical reactions, and I won't go into details about what exactly is going on, but one particular class of behavior that you might see is something that looks like spontaneous division, which is kind of interesting because these models are very minimal in the sense that they don't really include many detail, but you're already able to recapitulate some kind of interesting phenomena. So with regards to that, I kind of extended that previous model I was talking about to include the effect of some of these reactions not being in equilibrium, and also to see whether that might address some of these problems that I was discussing, such as the ripening of the droplets and the fact that larger droplets are worse than smaller ones. So I have kind of the same system as before, but I now have different sets of rules where the valency of the interaction, or once bound, once you kind of find once a specific reactant is formed, or once a specific product is formed, sorry, then the non-specific interaction is switched off. And that's kind of the only difference with regards to the previous model, and everything else remains similar. But let's see kind of, and this should be a video. So even when but now this model, because there's these kind of, there needs to be some kind of energy input to change these reactions, you already start to change some of the behaviors that you saw before. So in this example, you see similar kinds of dynamics as had existed previously, except now when you form a large droplet, it no longer is stable to the formation of product, because previously when a product formed, it would just stay in the droplet. But now, because you're switching these interactions, they get expelled from the droplet. And so what you see in terms of this, when you have a system where there's droplets and chemical reactions, but they're non-equilibrium, is you see this oscillatory behavior. I hope that the video is well played for you, where you form firstly one droplet, and then that droplet is not stable to the formation of product. So that droplet will start to kick out all of this product, which is labeled in red, and all the blue ones are just haven't found the product yet. And then kind of as you concentrate the gas from the level of before, you nucleate another droplet, and dynamically this process repeats again and again. So and this is just that kind of simple extension from the equilibrium model, including some reactions which are out of equilibrium. And I think this, you'll see again that these two will, eventually droplets will diffuse and then combine again, and then that will lead to an explosion of output again, of product again. So yeah, so slowing this process down, you can see what's kind of going on, which is when this droplet forms, and I have all of these kind of compatible reactants in there, and they start forming this product, which then gets expelled, which gives you an idea as to why this droplet is not stable. The droplet containing the full system is not stable. And then that process repeats. So already kind of, if I were to kind of, if you cast your mind back to that previous graph where there was evolution in time, or where I showed you kind of the ratio of product as a function of non-specific interaction, which was an equilibrium. Now the process is dynamic. So you see at these points where you start to form big droplets, you have these reactions occurring, which are you have these kind of brief periods of intense chemical activity, which is outputting many of these formed pairs or products. So in this way, you kind of have a extra dynamic timescales for tuning chemistry, and yeah, and not only that, but the final droplets are kind of become, these two different droplet states become chemically organized in a way that's different from random. So just by including, say, some non-equilibrium chemical reactions, you start to obtain kind of tunable, different timescales that can give you tunability over production of product. And you also you start to organize these droplets with respect to their chemical identity, which is no longer random. So in this one, you see, this is kind of a metric of how organized the droplets are, and the orange curve is the randomly organized droplet in terms of the makeup of all of its different components. And so this droplet, which is kind of half the size of the system, is much more organized in terms of its chemical identity than a random droplet would be. And yeah, you can kind of repeat this process for many different kind of let's say that the size of the system is scaled up, and now I start to see many different droplets forming, but there's no ripening in terms of these droplets aren't getting bigger in time. And in fact, what these droplets are doing is they're kind of constantly, they would be, in the absence of this non-equilibrium nature, they would be constantly producing, they would be constantly growing. But because of this non-equilibrium behavior, what they're doing is they're constantly, there's an outflux of product from each droplet. So they're acting like a chemical reaction center without having any of the problems associated with the equilibrium mechanism, which is interesting. But how it relates to biology is a kind of tricky question. So I'll skip this slide in the interest of brevity. So that's kind of my most recent work is kind of in vivo, that being able to prove or disprove such a hypothesis will be difficult, but using kind of technological platforms where some of these similar kinds of reactions that I've been discussing, which is the formation of say some specific pair and changing interactions are realizable using more DNA nanotech kind of approaches. Even though kind of so this is kind of a DNA nanostar, which people have been looking at with regards to these kinds of properties recently, which I've been modeling in a kind of abstract ways. So as in a difference to how I was modeling things before. But the idea being that some of these problems of some of these outputs of having combination of phase separation and chemical reactions might lead to these novel behaviors. So yeah, can we get some kind of tunable droplet behavior as a function of chemistry? So yeah, with that, I think I'm slightly over time, but I'll just conclude with some thoughts that spatially organizing chemistry could have benefits for cellular function. Obviously, it does have benefits, but spatially organizing chemistry in this method could have some benefits for cellular function. And the prospect of reactions out of equilibrium and coupled to droplet behavior has, I think, many potential interesting outcomes, which we're only just beginning to explore. So yeah, thank you. And I'm happy to take any questions.
Great. Thanks. That was super interesting. So you're going to be moving towards actually experimental work now?
No, so so I'm kind of moving towards a modeling platform which is much more closely resembling something that can be experimentally realized by someone else, because I have thus far, so another personal anecdote, I haven't done an experiment since undergrad, so I'm not in the position to be able to do something like that.
Great. Are there any questions?
I always ask questions, so I don't want to be the one who asks questions. Someone else.
Eric, we rely on you asking questions, but I know you have an interest in this topic too.
I know I do. It's one of those things where I know just enough to know nothing. You know, like I think I know something, but I don't. Okay, I'll go. So this is really cool, and I think it's extremely fascinating. One thing I wanted to ask you about, and this is me not knowing the experimental literature super well, but I think I've seen talks by people like Michael Rosen and those types of people with proteins that have ordered domains and then disordered linkers.
Right.
That seem to form stable phase-separated things where you don't have this phenomenon of ripening, if you will.
Yes.
And there's no chemical reactions in those. I mean, these are in vitro experiments. As far as I know, there's no ATP. They're not doing anything.
Yes.
And yet you get stable cases where the aggregate doesn't become the whole system.
Yes.
Do you have any thoughts about that? Is that interesting? Is that just kinetically trapped?
Yeah, so I would say that's usually, so that will be I mean, that process of ripening is a very slow one. So it goes as like a t to the 1/3 power, and some emulsions can be very stable kinetically over very long periods of time. So my understanding of when you see kind of many droplets, so either it's that, or if it's done on a surface, or if it's done in kind of 2 dimensions or something like that, the droplets might wet onto a surface, and you might not have the mechanism of ripening is some slow exchange between the gas phase and so on.
That's
so
the
it might
weird
take
thing.
a very
When they do frap experiments, though, those things frap fast, right? So when they do fluorescence recovery experiments, it's like on the time scale of minutes to get the whole thing recovered. So I find that super fascinating. But anyway, I think that what you're doing is really cool and going in the right direction. So that's really interesting.
Thank you. Yeah, I mean, with regards to other things, there might be some mechanism which is stabilizing surfaces anyway, not in a non-equilibrium regime. So, and I don't know if cells actually do this, but kind of industrially, we ourselves stabilize emulsions using different surfactants, right? And so that might also be occurring. Though I'm not entirely sure about
with these multivalent thingies, they don't add a surfactant, right?
Right.
But again, that could just be kinetically trapped. I don't know the literature. I don't know if they've waited long enough, you know.
Yeah.
But I find it a little. Anyway, very cool.
Great. Well, thanks again for a really fascinating talk, and finding new connections between different people in the community. So cool.